Hello everybody, welcome back to another STEM Upstarts video, and it's Kenneth, and today we're going to be kind of continuing on with what Abiram was talking about in the last video, because he brought up a lab that we performed in a class that had a three-step like process, and the bacterial transformation was actually the first step. So I want to talk about the second step, which is purifying or like obtaining the protein we want from the bacteria. But just to recap, you should go check out his video for more clarification. But also, I brought some stuff here. So um, he talked about this plasmid that we were putting into the bacteria. Basically, we were just introducing new genes to the bacteria. And our target gene, GFP, is on this plasmid. And that's a gene that glows like fluorescent green under UV light. So that's how we know like we have obtained it. And that's what we're going to try to purify from the bacteria or obtain from the bacteria. Here's the growth chart and just like the different types of plasmid, like minus PGLO means the bacteria don't have the plasmid, and then like the AMP is like the plate has ampicillin on it. So you can look at that, but if you want more information, please go check out Abram's video. The should be the one right before this. So how do we isolate this GFP, or how do you isolate any protein in a bacteria? Well, first you need to isolate the proteins, like all the proteins in the bacteria from everything else, like all the other cell components. So, and how do you do that? Well, so we would take our sample bacteria, which in this case, there were two plates containing the plasmid, but that was just the result of our lab. Just in general, you would just want to take the sample containing the bacteria that have the protein, and you would want to add them to tubes and culture them so that more bacteria is made. So basically, you're incubating it, or and you're getting more bacteria for use. If you already have enough bacteria for use, then you can skip that step. But we didn't have that much, so that's what we had to do. So after that, you're going to centrifuge the bacteria once. Um, and the reason you do that is to separate the bacteria from anything else. So in this case, the bacteria is like heavier, so it ends up in the pellet, which is like the solid at the bottom of a centrifuge tube. It's pretty easy to tell the difference between the pellet and the supernatant, because the supernatant is just the liquid in a tube. So um, everything that's not the liquid would be the pellet then. And the bacteria is in that pellet this first time. So we remove the supernatant from the tube because it doesn't have the bacteria, and obviously the bacteria contain the protein that we want to isolate, so we don't need the supernatant. And then an enzyme called the lysozyme is added to the bacteria. Why is this added? This is added because it breaks down the cell wall of the bacteria, which is useful because breaking down the cell wall and rupturing the bacteria, which the bacteria will rupture after you freeze it. And that leaves the cell components available. Like, okay, so they're available to get for us. So just to recap, you centrifuge it, the bacteria ends up in the pellet, which is the solid at the bottom of the tube. You add lysozyme so that the bacterial cell wall breaks down, and then you freeze it so that the bacteria completely ruptures, leaving only the cell components, which includes the protein we're looking for. So after that, you centrifuge the tube with the ruptured bacteria, which separates the proteins into the supernatant and all the other cell waste into the pellet. So unlike the first centrifuge where what we wanted was in the pellet, obviously we want the proteins, and those are in the supernatant this time. So we're going to keep the liquid. We're going to keep 250 shouldn't be milliliters, it should be microliters. But I can't really make that symbol on here. 250 microliters in our example of that supernatant, which contains the proteins, is added to a separate tube with 250 microliters of binding buffer, which I will talk about in a bit, to prepare the solution for the chromatography process. And the chromatography process is how we determine that we've isolated the protein. So first, what is chromatography? I don't have that description here, but what is chromatography? There's a liquid phase and there's a mobile phase and like a stationary phase, right? The stationary phase is already in the column, and in our case, this is a hydrophobic matrix. You can see here beads that have a hydrophobic matrix, like they form a hydrophobic matrix. And why this is specific for our case too, because the protein we want to isolate in our lab is GFP, and GFP is highly hydrophobic. And so since it's hydrophobic, we want the matrix to be hydrophobic too. Um, so so we add, well, you have the chromatography column with the stationary phase, which in this case is the matrix, the hydrophobic matrix that I just mentioned. And what you do is you add 2 milliliters of equilibration buffer into the column and let some drain out. This is just to, as the bullet point says, ensure that the beads do have the right hydrophobic properties for the rest of the lab. Um, and if you're doing a different protein, then the matrix or the stationary phase may be completely different. It could have completely different properties. It really depends on what you're separating and why. But this is just our general example here. So the bacterial cell components obtained from the prior lab, which is just the proteins, not all the cell components, but the proteins that we isolated. We said we added the binding buffer, which is this high salt concentration buffer mentioned here. So why is why is the binding buffer a high salt buffer, and why is it mixed with the bacterial, with everything in the supernatant, right? So a high salt buffer causes the hydrophobic amino acids in a protein to like twist and fold to the outside of the protein. And what this does is it allows the proteins to bind to the column or the stationary phase that is in our chromatography tube. So so basically you need the binding buffer to get the proteins to stick to the chromatography column. So the cell components in the supernatant obtained from the prior lab steps is mixed with this binding buffer and ran through the hydrophobic column. And this, as it says, it causes the proteins with the hydrophobic amino acids now on the outside to bind to the hydrophobic stationary phase or column. Next, then a medium salt concentration buffer is ran through to remove the loosely bound proteins. So these proteins are not very hydrophobic, which means they don't have many hydrophobic amino acids. So a medium salt concentration buffer kind of causes it to lose the strength that it had before because the other one was high salt. So now there's less hydrophobic amino acids interacting with the column. And since it's not very hydrophobic, that just kind of causes, if you imagine, the loosely bound proteins to just fall off and leave the column. And this can be a very complex process. I mean, if you just read what it says a couple times, it's a lot better to like do it yourself. And you can probably find videos of people doing this lab too. But So GFP stays on because it's very hydrophobic. So although maybe some of the hydrophobic amino acids will go back inwards, it's still very hydrophobic. It stays bound to the the column. Okay, and then lastly, water, which is also known as an elution buffer in this lab, has no salt. Okay, it's just water, which causes the hydrophobic proteins to like move or hydrophobic amino acids to move back inside the cell uh, or inside the protein, and that causes the GFP or any other highly hydrophobic proteins, I suppose, to unbind from the column, and that leaves it alone without the other ones into a uh, solution that we can use. So basically, um, through each step, you put the results in a separate solution. So um, now we just have the GFP alone in a solution. And since it's isolated, and you can test that it's isolated, if you shine a UV light on the solution, it should glow green, which shows that we've isolated the GFP. And since that's isolated from everything else now, um, you can move on to the next lab step, which is protein electrophoresis. And that's to confirm that you have GFP or whatever protein you're looking for. Because you can use molecular weights and stuff, but that'll be talked about in a different video, so I'm not going to go into that in depth now. So here, what I'll do here is I'll zoom out um, so that you guys can read this if anything doesn't make sense. Just keep in mind that this should be microliters and not milliliters. Um, and here it should be milliliters, not microliters. And so if you guys have any questions, let me know. I know this can be very confusing. And keep in mind that this is just for GFP, which is a hydrophobic protein. And if there's any other types of proteins or you're like trying to separate it based off any other difference, then there will be different processes or different types of stuff in the chromatography column that will be used. So just keep that in mind. Have a good day, everybody. Thank you so much. If you liked this video or found it helpful, please like and subscribe.